So we are off to a great start. We are in week 3 of class, so I'm very excited for that. We did do a homework assignment last week where you guys made a cell model. If you would like to share your models live with the class because you made a 3D model, we are going to start with that. If you turned your model in via Moodle, then I will go ahead and take a look. Well, I have taken a look at it, but I will go ahead and send you a grade. Anytime I grade something in Moodle, you should get an email saying that you have an item that has been graded. So is there anybody, just raise your hand, if you have a 3D model that you would like to share with us? All right, awesome. Looks like we have no one there. Then what I'm going to do is pull up some of the other files that I have so that you guys can share your models or your pictures with us. And as I am getting those situated, on your screen right now, there is a slide that has some bacteria on it. Now these are bacteria that are being seen through a microscope. What I would like you guys to do is to come up with, on the whiteboard, a brainstorm of what you are looking at on the screen. So for example, the shapes of them. Size isn't going to be possible because we are looking at a microscopic image. You could compare the size of them by saying the circular ones are larger than the rods, or whichever way you believe it works. But to say something is large does not work, but you can compare the size. So I have gone ahead and unlocked your whiteboards, and what I'm looking for is characteristics of the bacteria on the slide. So go ahead and put that up here for me. Whoops. Okay, you guys have some great things up here. Go ahead and add a couple of more. It looks like there's only 2 people writing. Doesn't have to be super scientific. I'm not looking for a master thesis on this. I'm just looking for your observations of the characteristics of the bacteria on this screen. All right, finish the one that you are working on. And this slide is actually a combination of lots of different types of bacteria. None that I actually even know the name of, other than to say it is different types of bacteria. So let's look at your characteristics and your observations and see what you guys can say. We have some of the bacteria looks like string and some of them look like dots. Very good. That is because there are different types of bacteria. Different types of bacteria have different shapes. There are three main shapes though. It has string-like structures. Very good. There are some spaces where there are a lot of bacteria and some spaces where there are not a lot of bacteria. Perfect. Some of the bacteria are shaped like long rods. Others are circular and oval shaped. It wraps around and almost looks like spaghetti. Interesting. The bacteria also seem to be floating in some sort of liquid. Some bacteria have been colored pink, whereas others have been colored a dark purple, probably for them to be seen better. And it looks like silly string. Well, those were some great observations, and definitely some observations that we will kind of look in more detail at today. There are three different types of bacteria. That is why there are three different shapes of bacteria. These bacteria have been colored. They have been colored with indicator dyes, and that is to make them easier to see. If you were looking under a microscope and you were looking at just a slide with uncolored bacteria, it is very difficult to see the bacteria. They are small, and their walls are very thin and hard to see. If you look at a cell underneath a microscope, it is much easier to see. So scientists do dye bacteria. They do this with something called a gram stain. We are not going to be gram staining anything today. We are actually not even going to be looking at anything live under a microscope. I explained that to a couple of you before class started, but in order to get a good microscope, they are quite expensive, so it is not something that we require you to have for class. If you do have a microscope, let me just grab something here. If you do have a microscope, there is a container of prepared and blank slides in your lab kit. You certainly are more than welcome to look at the slides. Just remember, they are made of glass and they are quite fragile. Okay, so we are going to start off by looking at Gabrielle's plant cell, or I'm sorry, animal cell project. So Gabrielle, I put it up on the document screen. Do you want me to leave the picture up? Like, do you have the written words somewhere else, or would you like me to read the words up so you can refer back to it? And just tell us about your project.
I don't have the words with me right now, so you could. Okay. The ribosomes are the tiny blue dots spread across the animal cell. Ribosomes are small organelles that are sites of protein synthesis. The lisosomes are the yellow and black circles in the animal cell. The digestion of cell nutrition take place here. The mitochondria is the orange, green, and yellow peanut-shaped figures in the cell. The mitochondria convert the energy stored in the glucose into ATP, adenosine triple for the cell. The DNA deoxyribonucleic acid is the middle of the nucleus, and it is red with black spots inside of it. Ribosomal RNA is produced in the DNA deoxyribonucleic. The Golgi apparatus is the blue and black organelles towards the left that look like stacked pancakes. The Golgi apparatus packages proteins and carbohydrates into the membrane-bound vessels for export from the cell. The nucleus is located in the middle of the cell, and it controls many of the functions of the cell. The smooth endoplasmic reticulum is the pink organelle coming from the nucleus. Smooth ER transports materials throughout the cell. The rough endoplasmic reticulum is the pink organelle coming out of the nucleus with purple dots on it. The rough ER is covered with ribosomes that give it a rough appearance. Rough ER transports materials throughout the cell and produces proteins in sacs called cisterns. The cell membrane is the outer shell of the cell. It allows some substances to pass into the cell and blocks others out. The cytoplasm is the white surrounding all the organelles. It is where the organelles are located.
Perfect. Thank you very much. And this is the diagram that Gabrielle went ahead and put up for us. She did a fantastic job, I believe, with her drawing. Way better than anything I would to do. She has her key on the right-hand side as well as her pictures on the left-hand side. So great job, Gabrielle. Thank you very much for sharing. Okay, next we have two images. And we are going to pull them up. For some reason, they're not coming through on the whiteboard. So what I'm going to do is I'm going to pull them up on my desktop to share with you guys. So you're going to have to bear with me for 1 minute as I pull these up. And okay, Sarah, yours is the first one up. I just need to give you control, and then I will bring it back up. So Sarah, go right ahead.
I made an animal cell, and the pink middle round circle is the nucleus, which holds DNA, and it includes the nuclear envelope and the nucleus. And the green oval-shaped things, that's the mitochondrion, which generates energy for a cell to function, and it is often known as the powerhouse of a cell. And the circular brown is a lysosome, which digests and recycles excess organelles and food particles. And the yellow towards the bottom right, is a centriole, which helps the cell when it is time to divide. And the orange, kind of in the middle, is a Golgi body, which takes fats and proteins from rough ER and modifies them into vesicles. And the blue, that's right above it, kind of, is smooth endoplasmic reticulum, and that is a storage organelle, and it stores steroids and lipids. And the dots kind of below it are free ribosomes, which make proteins in the cytoplasm. And the blue that you see in between all of those is the cytoplasm, which fills the cell with fluid, keeps the organelles in place, and it uses enzymes to break down large molecules so other organelles can use those products. And the kind of rim that you see that goes around that is the plasma membrane, which has many functions. One function is that it separates the contents of the cell from what's outside the cell. And the coat that you see that's blue is a cell coat which covers the plasma membrane. And that's it.
Awesome. Great job, Sarah. Thank you very much for sharing that with us. The next one that we have is Alicia's. So Alicia, same thing. I'm going to give you the microphone and then I will share it with you guys over the desktop. So Alicia, go right ahead.
Okay. Let's see. I guess I'll start with the rough endoplasmic reticulum. It folds and produces some proteins and is involved in controlling quality and release. It's the one studied with the ribosomes, and that's why it's called rough. The smooth endoplasmic reticulum regulates and produces fats and steroid proteins, and it's smooth because it does not have ribosomes all over it. The mitochondria is the powerhouse of the cell, and it helps produce power and stuff. The nucleus is kind of like the brain of the cell. It helps regulate instructions and gives kind of like a purpose to the cell. Ribosomes synthesize protein and translate code for RNA production, so it helps with the production of proteins. The cell membrane is the outlining of the cell that protects from invaders and regulates passage into and out of the cell. This can be through passive transport, active transport, and so on. The lysosome maintains homeostasis within the cell. It helps regulate so that way you know that the cell doesn't just blow up, I guess. The Golgi apparatus is the storehouse of the cell. It helps package and export things into the cell and just get things around. The cytoplasm is enclosed within the membrane. It is a thick solution that contains all the organelles for the cell. And the DNA deoxyribonucleic acid is the coating of the cell. It's the memory of the brain. It helps make DNA kind of like our DNA, but it helps make the nucleus what it is. You know, it's an animal cell or a plant cell, I guess you could say. And yep. And then there's the diagram and magnification.
All right, fantastic. Thank you very much to everyone who shared today. Did anybody else have a model that they wanted to show me over the video camera? I know one of you came in late. I did not get any other homeworks, so if you think you turned it in and I did not just share it, please give me a call after class today so we can figure out why it didn't go through and get it corrected. And if you didn't turn it in, reminder that your homework is due by 9 a.m. on your class day. So it was due at 9 a.m. today. Every day you are late, so anytime after tomorrow at 9 a.m., you lose points. Okay, you lose 10 points. So basically you're losing a letter grade for every day you are late. This is a class where you do have the potential to earn high school credit. You need to complete assignments on time. Okay, for several reasons, one of which being we are going to use what you did last week and build on it this week. Okay, so make sure homework assignments get into me on time. Those of you who did submit them, awesome job. I was impressed with them all. Now that I have seen your presentations, I will go ahead and assign a grade shortly after class today. So great job on those. Today, again, we're going to talk about bacteria. We are going to actually do two different labs today. One of them is going to build a little bit on the scientific drawing that you did last week in class and that you practiced with your animal cells or plant cells that you were to create for me. And our second lab is going to be a lab that we set up today and that you continue and observe during the next week. Before we go ahead and do either of our labs, however, we are going to take a look at some information about bacteria in a PowerPoint. Now remember, I promise this is nothing like last week. So today we are going to look at bacteria. What is bacteria? Well, there are several characteristics of bacteria. The cells of bacteria are prokaryotic, and they are among the smallest known cells. They have no membrane-bound nucleus. They have ribosomes, but no other organelles. And their DNA that is present as long circular molecules. Where are bacteria found? That's partly what we are going to do in our second lab today. We are going to look at different habitats near you that you have access to, either indoors or outdoors, where bacteria is found. But bacteria is found everywhere. It is found in the air, it is found in water, it is found in soil, it is on plants and animals, and it is found in plants and animals. There are three major groups of bacteria based on their shapes. There are coccus, bacillus, and spirillum. We saw all three of these in that very first slide I showed you at the beginning of class. Coccus bacteria is shown in these two scientific drawings. Now, if you look at these scientific drawings, they're inside a circle, which indicates that they are being looked at under some sort of magnification from either a microscope or a magnifying glass. The plural of coccus is cocci. Coccus bacteria are spherical bacterium. They look like little spheres. There are different types. There are monococci, which are single cells. So that would be one sphere by itself. There are diplococci, which means there are two spheres connected together by themselves. And there can be streptococci, which means it is a chain of spheres connected to each other. The next type of bacteria is bacillus, whose plural is bacilli. Bacillus is a rod-shaped bacterium. Bacillus exists as single cells, in pairs, which are diplobacilli, and in chains, which are streptobacilli. And the third type is spirillum, whose plural is spirilli. Spirillum are a spiral-shaped bacterium, and they exist only as a single cell. You cannot have chains of spirilla or pairs of spirilla. They are just one single cell. So let's look at the composition of a bacterial cell quickly. Bacteria have a cell wall, which is the outermost structure of the cell. You can see it in the diagram to the right. Right inside of the cell wall is a cell membrane, and that cell membrane in bacteria is made up of peptidoglycan, which is a long chain of sugars linked to amino acids. Penicillin destroys bacteria by interfering with the peptidoglycan molecules. Okay, penicillin is a type of antibiotic used to destroy bacteria. And the way it does it is by attacking the cell membrane. Cell membranes are found just beneath the cell wall. They may be folded inward, and they have an ATP. Bacteria produce ATP or energy that takes place in the folds of this membrane. Bacteria are encoded or encapsulated in a capsule, which is a layer of slime secreted over the cell wall of the bacteria. This capsule provides protection for the bacterium. Bacteria that have this capsule are said to be encapsulated. Most pathogenic or disease-causing bacteria are encapsulated. Bacterial structures have flagella, or may have a flagella. Some bacilli and spirilla can move like by using a flagella, which is a whip-like structure. And if you look at the picture on the right here, you can see a bacteria with flagella. Okay, and like all living things, bacteria does need food, which is why when we make our Petri dishes today, we are going to put nutrients in the Petri dish for the bacteria that we are going to hope to grow. Bacteria can also be categorized not only by their shapes, but on how they get their food. The two categories are heterotrophic and autotrophic. Heterotrophic bacteria get their food from a source of organic matter. They can be saprobes, which means they feed on dead plants and animals, or they can be parasites, which means they live on or in the organism and cause diseases. And autotrophic bacteria make their own food. There are some autotrophic bacteria that are photosynthetic, which means they use a special type of chlorophyll called bacteriochlorophyll to produce their own food from the sun. However, oxygen is not released in bacterial photosynthesis. They can be chemosynthetic, which means they obtain energy by breaking down inorganic materials such as sulfur. Generally speaking, bacteria grows when it has food. However, there are some factors that limit the growth of bacteria. Those include temperature, pH, moisture content, nutrient availability, and competition from other organisms. And when the bacteria do reproduce, they reproduce asexually by binary fission. And generally speaking, if you were to take an average, bacteria reproduce about every 20 minutes. And that is where we are going to stop in terms of slides today. What we are going to do now is we are going to look at lab number 11 in your lab book, which is on page number 49. Now on page 49, it talks about bacteria, the bacillus, the coccus, and the spirillum, and the difference between diplococcus and streptococcus. Now, this actually asks you to look at slides under a microscope. Well, since I told you I don't require you to have a microscope, I'm going to go ahead and pull up some microscopic images on the whiteboard for you. And we are going to start with the first. Okay, this is the first slide we are going to look at. Now, if we look at the procedure of our lab, it says examine carefully the photos of each type of bacteria, Coccus, Bacillus, and Spirillum. Notice differences in size and shape. All images have the same zoom level of approximately 19,000x. So the pictures on page 50 and 51 are all zoomed to the same magnification. So we can compare size. Now, does anyone want to make a comparison between the three images on 50 and 51 for me? Just go ahead and turn your mic on and start talking. Maybe which one seems to be larger? Go ahead, Alicia.
The coccus, you can see how the coccus and the bacillus, they are grouped at some points, but the spirillum is not. It's just separate, like you talked about earlier. The coccus also seems to be smaller compared to the bacillus and the spirillum, and the spirillum seems to be the largest compared to the two. There seems to be less spirillum in one slide than in coccus or bacillus as well. And I do believe that's all I got.
All right, fantastic. Thank you very much for sharing. You definitely can see some of the Coccus and Bacillus bacteria in pairs and in chains. And you can compare the sizes as well. So fantastic job with that. Okay, on the whiteboard, if you take a look, you can see some Coccus bacteria. And I'm going to put another image up next to it of Spirillum. And a last image, nope, a last image of Bacillus. Let's get the other one up there. So these are just three other examples where you can see the difference in shapes. And again, if you look at these images closely, you can see the pairs in the Coccus and the Bacillus, and that the Spirillum have no pairs connected to them. Now, what I'd like you to do is flip to page 54 of your lab book. Alicia pretty much answered questions 1 through 3 when she went and did her little review. So now we are going to draw and label what we see. So I'm going to put a slide up on your whiteboard that is mixed bacteria. And there we go. We'll use the same image we used in the beginning. So here we have a slide with mixed bacteria. On page 5, there are 3 circles. Number 5 says, there are several drawing circles below. Make a drawing of Coccus, Bacillus, and Spirillum bacteria you may find. You may also use all of the circles provided or just draw them all in one circle. If you do the latter, make sure you label the bacteria and groupings you find. Also, title the circle appropriately. Bacteria instead of coccus, etc. Okay, so what we are going to do is you are going to, in one of these circles, or in all three of these circles, you are going to draw examples of Bacillus, Spirillum, and Coccus bacteria. I would like you to make sure you include Diplococcus and Staphylococcus and Diplobacilli and Staphlobacilli. And label your drawings. I'm going to give you about 5 minutes. These do not need to be super fantastic drawings, but what we are going to do is you are going to share them each with me, or share them with me, so that we can look at them. So you're taking 5 minutes to draw Staphylococcus, Diplococcus, regular coccus, Bacillus, and Spirillum bacteria on page 54.